Yo, yo, yo. Welcome to How to Photosynthesize with Mr. Tran, credited by Kurtz. So we know that photosynthesis is a redox reaction. That's right, redox reaction. And when we see that word redox reaction, we know that a reactant is being oxidized and a reactant is being reduced. And one way to remember which is which is to use this mnemonic device. Leo says GER. So losing electrons is the LE there, is oxidation. And then gaining electrons is reduction. So instead of calculating out that charge or the formal charge, we are simply going to be looking at the reactants and the products and looking for the change in H. So again, this is my tip for you guys. Always look for the change in H. Look for the change in H. And so let's actually write out that chemical equation. It's 6H2O. You're going to see why I start off with H2O. 6CO2, and you're also going to see why I am using the balanced chemical equation. We have light, and then forming glucose, and 6 oxygen molecules. So, on one side we have the reactants, the other side of the arrow, the products. I'm underlining water, and we're going to look for that change in H. So when I am following, trying to predict what water becomes, we're looking for that change in H. And so the change in H would be not having an H since the reactant had the H. So this is losing the H for hydrogen. And so this is going to be oxidation. Now, we have CO2 over here. CO2 looks like it didn't have the H, and then it got the H after. And so we're gaining H, which results in the reduction. So again, needing to know which reactant is reduced or which reactant is oxidized is definitely going to be on your test. So get this down. So we have the chloroplast. We need to know the structure very, very well. Okay, so again, the chloroplast. The three structures that's needed would be, first one, the thylakoid. The thylakoid, again, captures that light, specifically on the thylakoid membrane. The second part we need would be the stroma. This is the aqueous fluid inside the chloroplast, and you can think of it as the cytoplasm, of the chloroplast, just like the cytoplasm is for the cell, gel-like fluid. And then the last one we need to know is the thylakoid lumen, which is inside the thylakoid, and then we can also call that the thylakoid space. Space and lumen can be interchanged. In order to understand the details of photosynthesis, we need to know the big picture first. So, photosynthesis overview. We know it occurs at the chloroplast. Always, always, always think in terms of the reactants and the flow of electrons. So that's why I'm writing out the equation down here to keep sure that we're paying attention to, the reactants, and then the electrons. So I'm going to note that up here. Always note what is happening to the reactants, and then the electron flow. Then we will understand photosynthesis very easily. Okay, let's begin. We have two sections that we see. One that has light, and guess what? It's called the light reactions. The light reactions takes place at the thylakoid membrane. It uses light to break apart water to become oxygen, so water is being oxidized, to create these high energy molecules here that feeds to the second process. So the two high energy molecules, chemical molecules, would be ATP and NADPH. And then the second step or phase of photosynthesis would be the Calvin cycle. It takes CO2 and fixes it in the stroma. When it gets fixed, y'all, it's placed on glucose. So this requires a lot of energy. And I like to think of Calvin as the bad dude. He uses all of that ATP, high energy, and the high energy electrons from NADPH to again fix carbon dioxide to glucose. Well, once all that energy is gone, guess what? Calvin kicks those used products back to the light reaction to get more energy. So Calvin takes. Okay, so let's spread out. NADP plus and NADP in the inorganic phosphate is kicked back to the light reactions. So you might be asking, Mr. Tran, what is NADPH? Well, NADPH and NADP plus are considered to be electron carriers. Since electrons cannot freely travel without the interactions on molecules, it needs to be safely carried to the desired reaction. So these guys are considered to be the carriers or vehicles in which they transport electrons to a specific reaction. So that's what NADPH, NADP plus are. Then my second question to you is, can the Calvin cycle work without light? And that is no. Light drives these high energy molecule creation to be fed to the Calvin cycle. So the Calvin cycle needs light, but the key is the Calvin cycle doesn't utilize that light directly. It is independent of light. Okay, so does not work with light necessarily, but depends on the products of the light reaction in order for it to continue. Okay, we're going to look at the very specific steps of the light-dependent reactions. First off, when we're looking at this complicated diagram, we need to be able to label all the parts. So that's what I'm going to do. I'm labeling the thylakoid membrane. And then the thylakoid membrane separates the thylakoid space or lumen from the stroma. And then now let's label the major components. I'm going to do this in blue. The major components of the light-dependent reactions are embedded in the membrane. The first one would be called the photosystem. They are numbered. This one is called Photosystem II I. We'll get to that later. So a photosystem, again, is a collection of proteins and pigments. So these are called chlorophyll pigments. They, again, pass on that light energy to this very important chlorophyll. So I'm going to label that now. It's called chlorophyll A. So the second component of the light-dependent reaction would be the ETC, or electron transport chain. The electron transport chain is just a collection of proteins that pass along the high-energy electrons. Okay, so it allows the electrons to move across the thylakoid membrane. Now, we approach our third structure. It's another photosystem. Again, collects light, but this one is called 1. The reason why this is 1 is because the names were given based on their time of discovery. They found Photosystem I first, but realized that Photosystem II actually goes before Photosystem I, so it's backwards. Then our next component is this circle here. This circle is an enzyme called NADP plus reductase. That NADP plus reductase is important. And then finally, we have arrived at the last component, and it's called ATP synthase. Okay, with that said, I'm labeling everything else in green. So I have broken down the light-dependent reactions into 5 steps. Okay, 5. So the first step is easy. Light is captured by the photosystem II, and then the pigments pass that light on again until it gets to chlorophyll A. Chlorophyll A's electrons gets excited by this light energy, and it goes here. So that's the first step. Light excites electrons in chlorophyll A of photosystem II. The electrons, they move to ETC. And I'm going to pause here and explain the second step. So when water excites that electron, it is now considered to be empty. Okay, so there's no more electron here. It's empty. It got excited. Well, photosystem II enzymes over here, well, they will split water apart. So water is split into oxygen, and this, yes, y'all, this is the product, the byproduct, okay, escapes through the stomata. Then hydrogen ions are also created, which contributes to this here inside the lumen. And finally, it releases electrons, and the electrons are placed back on chlorophyll A. So the big question is, where is what is the source of the electrons that are moving all the way to the Calvin cycle? Well, it comes from water, so you need to know that. Okay. Now, let's write out let's rewrite that. Let's talk about the enzymes. So, enzymes on photosystem 2 splits H2O to create O2, H plus, and electrons. Well, the O2 is the byproduct. The escaping through the stomata, right? The H plus contributes to the concentration gradient inside the lumen, and electrons back on chlorophyll. So we have the two steps there. Let's continue. Well, the electrons again are here. The electrons are very high energy, and as it moves through the electron transport chain, it gets to be low energy as it is accepted by chlorophyll A on photosystem 1. So there's that loss of energy, but remember, the law of thermodynamics states that energy cannot be created or destroyed, simply transformed. And again, this energy is being utilized to do this. It takes a proton that's considered low concentration and pumps it into the lumen, which is considered to be very high concentration. So going against the concentration gradient using a proton pump requires energy, and that energy lost from the electrons being transferred across the electron transfer chain powers that proton pump. So we can call this the proton pump. So that's the third step. The third step is important. So you can pause to write the two steps down. I'm going to be erasing it. And again, writing step 3 out of our 5-step process. So, erasing. Okay. Now, let's write out step 3. High energy electrons move through the ETC, losing energy along the way. That energy drives a proton pump to move H plus from low to high. We're going to label that. That's stroma to lumen. Electrons end up at photosystem 1, specifically the chlorophyll A. You may ask, is that the same chlorophyll A? No, you'll learn about all of that in college or in advanced classes, but those two chlorophyll A's, they have different names for them. But we won't dive into it here. So let's continue. Now we have our fourth step. The fourth step is, yep, you guessed it, the process of light being gathered by the pigments, passing along until it reaches chlorophyll A at photosystem 1, exciting that electron. That electron is immediately carried to NADP plus reductase, that NADP plus reductase adds that high energy electron to this molecule here called NADPH. It does this by combining NADP plus and a hydrogen ion to make and capture that high energy electron. Okay, so this high energy electron carrier now goes to the Calvin cycle. So let's review that in our steps here. The fourth step. Okay, fourth step. We're almost done. So light excites chlorophyll as electron in photosystem 1. Electrons are placed on NADPH by NADP plus reductase. NADPH goes to the Calvin cycle. Last step. Well, this has been building up for a while. We had contributions of hydrogen ions from the splitting of water. We have contributions of hydrogen ions being pumped into the lumen from the movement of the high energy electron in the ETC. And it builds up right here, a collection of high or high concentration of hydrogen ions. So that concentration actually diffuses across this enzyme, ATP synthase. So this is high to low. And so that doesn't require energy, y'all. But what that does is generate ATP. And so that's this vocab word. When protons are diffusing across the proton carrier or this ATP synthase, it generates ATP. This term is called chemiosmosis. And how it does it is that the movement of the concentration of hydrogen ions across the ATP synthase turns the motor here, capturing ADP and inorganic phosphate to generate ATP, which goes to the Calvin cycle. So let's do that. We have number 5. So 5. The high concentration of H plus in the lumen diffuses through ATP synthase to create ATP, and that's called osmosis. The ATP goes to the Calvin cycle. There we have it. We have the light reactions. Light being transformed into these high energy molecules at the end right here and right here. So the last step is called the Calvin cycle. This happens in the stroma. We also need to note that the Calvin cycle is also called the light independent reactions. Okay, so where do we start? We will start with 3-RuBP, a 5, count it, 5 carbon sugar. It again gets combined with 3CO2 by this very amazing enzyme, RuBisCO. So I'm going to label it right here, important enzyme, to fix carbon to RuBP, and eventually that first step finally creates a 3-carbon sugar. This 3-carbon sugar, you don't need to know the name, but the name is there for convenience and understanding. 3-PGA, you can think of it as a low energy 3-carbon sugar. This first step is called carbon fixation. When you hear carbon fixation, it's simply taking something that's inorganic, like CO2, and incorporating it into an organic molecule. So this inorganic molecule, CO2, is being incorporated, combined with RuBP, split, and turned into 2, and mathing wise, 6, 3-PGA. So again, just think of it as 6 low energy carbon or 3-carbon sugars. So the second step is to turn this 3-carbon sugar into another 3-carbon sugar, but the big difference is it is high energy down here. So G3P is a 3-carbon sugar that's very high energy. So we're going to call it high energy. It actually creates 6 G3P in a step called reduction. So reduction utilizes ATP and NADPH. It uses the high energy in order to power this transformation from low energy to high energy. Well, with the 6 G3P, you need to know that name G3P. That 6 G3P, y'all, one of the 6 goes to make glucose. But Mr. Tran, glucose is a 6-carbon molecule. Well, you're correct, Billy. Um, how many G3Ps are needed to make glucose stuff? The answer is 2. 2 G3P equals 1 glucose. So we need 2 of these cycles to run in order to make 1 glucose. Okay, very good. Good question. So, how many CO2s are required to make glucose? That is 6. Let's rewrite this equation here. 6H2O and 6 right here, CO2 yields 1, 1, 1, 1, 1. Oh, nope, the video's not broken, y'all. I'm just repeating that number because, again, it all math out. Okay. Now, the other 5 G3P are recycled to regenerate RuBP. And in the, very, very, very classic way of naming that step, we're going to call it regeneration. In order to transform these 5 3-carbon molecules into 3 5-carbon molecules, ATP is needed, energy is needed. So the other question that we have to ask ourselves is, which steps in the Calvin cycle requires energy? And that will be reduction. That's a transferring of the energy to a low energy 3-carbon sugar and making it into a high energy 3-carbon sugar. And then finally, the changing from 3 to 5 requires energy. So regeneration is the other step. Cool deal, y'all. That was a lot. Hope that you guys understand the process. If not, rewatch it. Send Mr. Tran an email for any questions you may have. Have a good one.